Hello, everyone. I welcome everyone to join our RNA Collaborative Seminar Series. So today is our time from the University of Science and Technology RNA Institute to give the talk. We have two presenters. The first one is Dr. Ge San. Dr. Ge San is a professor in School of Life Sciences from the University of Science and Technology of China. He got his PhD at Georgia State University and did his postdoc at Emory Institute University and then at Yale University with Sid Adams. After that, he joined USTC as a faculty member in 2010, that's 12 years ago. And his lab has been studying non-coding RNAs for more than 10 years. So especially he focused on non-coding RNAs and circRNAs. So let's welcome Ge. It's your turn. Can you hear me?
Okay, so it's nice to have this chance to talk about circRNAs and especially I just share my PPT first. Okay, is that okay?
That's good. Yeah, it's working.
Okay. Here I will introduce some studies in our lab about circRNA, and also basically give the audience a brief introduction about the circRNAs. So, okay, basically in 1976, in PNPase, it's reported that in plant cells, something called viroids, circRNAs, actually they are single-stranded, covalently closed RNA molecules. Okay, this is the first image of circRNA that's published in this paper. So you can see in the microscope, the electron microscope, you can see they have this closed circular form. So from that time, since like more than 40 years ago, like people start to realize that RNAs can be circular, not just linear forms. So then like viroids and a single-stranded RNA viruses, and sometimes like during the tRNA process, you can see some circular molecules. So, but today, when we talk about circRNAs in like animal multicellular cells, basically they have this kind of four subclasses of circRNA, like exonic circRNA formed by exons, and exon-intron circRNAs formed by exon and intron. And also intron can form circRNAs. That's if the lariat, intronic lariat get partially degraded, it can generate cirRNA. And recently, circRNA encoded by the mitochondrial DNA also found, which is called MecciRNAs. So basically, this is the intronic circRNA, and then for circRNA, exonic circRNA, and exon-intron circRNA are formed by back splicing. So during the canonical linear splicing, the precursor RNAs actually spliced into amine. But in back splicing, it will generate a circular RNA. And actually, this kind of circular RNA were noticed by like a number of studies, like during decades. But as they are called like scrambled exons missplicing, and most of them are believed to be like almost splicing noise. It's like, but like in 2012, this paper published in PLOS ONE actually point out that hundreds of human genes actually produce circular RNAs. And very quick, in 2013, two papers published, they shown that exonic circular RNAs formed by back splicing of exons, actually they function as circRNA, which are microRNA sponges that can interact with complex in the cytoplasm and actually have physiological functions. So actually our lab started to get into the circular RNA field. Actually, we was investigating the RNA polymerase II for its transcriptional regulation. Actually, we put down RNA polymerase II, and then we sequence the RNAs that isolate together. We found more hundreds of circular RNA actually can interact with RNA polymerase. And then we characterized this circular RNAs, and we found actually instead of like exonic circular RNA that localized in the cytoplasm, this circular RNA actually localized in the nucleus. Those are the mRNA signals primarily localized in the cytoplasm. But EcRNA, the circular RNA we found actually EcRNA. We found they are composed of exonic and intronic sequences. So we call them EcRNAs. They are localized in the nucleus. So, and then with a series of experiments, we actually found out that EcRNA recruit U1 SNRP complex, okay, with its exon-intron splicing site. So then the U1 SNRP complex will regulate the RNA polymerase II and the gene promoter actually increase the transcriptional initiation of the parent gene. So this paper is already published. So basically from back splicing, we now know like exonic circular RNA and exon-intron circular RNA can be formed. Actually, I already talked about some circRNAs can function as microRNA sponges. Actually, there are also a lot of publications now showing that circRNA some of them can also be translated into polypeptides. So just recently, like last year, we actually published a paper that we found out that the mitochondrial DNA can also encode circular RNAs. What we have done that we isolate mitochondria from animal cells, mammalian cells, and then we sequence the RNA in the mitochondria, and we by bioinformatics analyze, we found out that mitochondrial DNA actually encode hundreds of circular RNAs. We call them MecciRNAs. So these are two examples by Rosenblatt. So for one MecciRNA, we found out that they actually interact with this protein, and if we by high resolution microscope, we can see the localization of this messy-RNA, the purple, and then the protein is injected, they can co-localize inside the mitochondria. Actually, if we look down the messy-RNA, and then we can see the protein that interact with this messy-RNA also show the decreased level inside the mitochondria. The total level of this protein is unchanged. So here is another example. So the messy ND5 actually interacts with HMPA1. Same similar results. If we lock down messy ND5, the interacted protein actually decreased inside the mitochondria. Also, we shown that messy-RNA interact with components like TOM40, which is the for transport protein into the mitochondria. Also interact with like PNPase, which have been shown to interact with RNAs that get transported inside the mitochondria. So this leads us to believe that actually messy-RNA facilitate the mitochondrial protein transport from the cytosol. So this is an in vitro experiment in this in vitro translational system. If we add messy-RNA and mRNA together into this translational system, actually we can see the messy-RNA actually increased the newly synthesized protein, the export of newly synthesized protein into the mitochondria. So this is messy ND1. And for another messy-RNA, same thing. So if we add the mRNA and messy-RNA together into the translational system, so we can see actually more newly synthesized protein get transported into the mitochondria. But if we add messy-RNA later after the translation of mRNA, so actually same amount of protein added into the system, then we can see now the messy-RNA almost cannot facilitate the transport of protein. So in this example, if we add messy-RNA later, the protein cannot get into the mitochondria. So this suggests a model that actually messy-RNA can facilitate the export of newly synthesized protein in the cytosol and actually promote the mitochondrial export by interacting with like TOM40 complex. So and this paper is already published. Last year we also studied a conserved exonic circRNA. So this conserved circRNA is encoded by the gene called Boule. Okay, this gene actually functions in the sperm genesis. So actually this circRNA is conserved from Drosophila all the way to human. So actually we examined fly, chicken, pig, mouse, and human. We found out the circular, we call it circBoule. circBoule RNAs actually can be found from fly all the way to human, and in mammalian cell, in mammals actually is especially expressed in the testes. So in fly, if we lock out this circRNA by delete the intron that involved in the circRNA biogenesis, we examine the phenotype. So interesting, we find out that under heat shock, the male fly actually, the fertility of the male fly actually decreased. Okay, in the lockout mutant, and this phenotype can be rescued by if we add back the circRNA. So what we found out is actually without circRNA, the fly sperm, the Drosophila sperm actually show a lot of defects and some cell deaths. So by RNA pull down, we found out that these circBoule RNAs interact with heat shock proteins like HSC4 and HSP60C. So further experiments, we show that these are actually in vivo experiments. We actually generate the in vivo tagged heat shock proteins, and then we can show that actually they interact in the fly testes. So further, we found out this interaction is mediated by two conserved RNA sites in the circBoule RNAs. So if we delete them, the heat shock protein will not interact with the heat shock protein. So this circBoule RNAs actually promote the degradation of this both heat shock proteins. If without circBoule RNA, we always see heat shock protein levels actually higher, especially under heat shock condition. Actually, we found out circBoule RNAs promote the polyubiquitination of both heat shock proteins. And in mice, actually the two binding sites also conserved. And we also find out circBoule RNA in mice actually interact with a heat shock protein. It's called HSPA2, which is also HSP60 family member. So this is an image of sperm of mice sperm. So we can, the red signal is circBoule and the green signal is the HSPA2 protein. Actually, they co-localize in the sperm. So by CRISPR lockout, actually we generate CRISPR lockout mice that without the circBoule expression. We can see that under heat shock, the mice actually mutant mice, the sperm of mutant mice actually show decreased level of fertility. For when we examine the in vitro fertilization ability. So here we can see at the molecular level again, circBoule RNA in mice also promote the degradation of HSPA2 protein by promote its polyubiquitination. So, and also interestingly, the mice circBoule RNA can rescue the fly defect. So in human, we also examined, we found out that human sperm also express circBoule RNA. And the two binding sites also conserved in human circBoule. If we delete it, then this interaction between the human circBoule and HSPA2 is abolished. And in people with ascendospermia, so which has weak sperm, so we can see that the circBoule level actually decreased significantly compared to normal sperm. So also in the patient, actually we can see HSPA2 protein has no lactive relationship with circBoule RNA. But in the normal sperm, actually we can see the circBoule level and HSPA2 level actually are lactive correlated. So actually what we found is a conserved circRNA that's conserved for like 600 million years from fly to human, and it's actually promote the male fertility to against heat stress by promoting the polyubiquitination of heat shock proteins. So this paper is also already published and get commented by like science and also faculty opinions. And very recently, we reported another circRNA. Also, it's a exonic circRNA called circRN1, which is highly expressed in the gastric cancer. And we shown that this circRNA actually can interact with the proteins called H9PM, which can regulate the oxidative splicing like VEGF-A. So to regulate the metastasis of gastric cancer. So actually today I talked about 4 major subclasses of circRNA in animal cells. So circRNA basically, this is a recent review that we published. circRNA now are found to like regulate development, stem cell, and like fertility. What I already introduced, like circBoule RNA, metabolism and aging. So and also it can play roles in like a series of diseases, like circRNA1, like in gastric cancer. Like now, circRNA are found to play roles in like metabolism diseases, like cardiovascular diseases, neurodegenerative diseases. So, and basically, I talk about like circRNAs, EcRNAs, and MecciRNAs. Pretty much these 2 subclasses are identified by my lab, and so that's I talked more on these 2 subclasses. And like at molecular level, like EcRNA promotes the transcription, and like circBoule RNA actually function as a protein scaffold, and also like circRNA1 actually function like a protein decoy to regulate the function of RNA binding proteins. So in summary, today I talk about 4 subclasses of circRNAs in animals. So basically, I will not repeat what I already talked about, but I just want to make a brief 1 sentence like summary is like circRNAs are actually very diverse, showed a great diversity. So in subclasses, functions, and functional mechanisms. So with this, I would to thank the like funding agencies for giving me the money for the study, and also thanks for like all the first authors and also collaborators in the studies that I talked about. Okay, and thanks.
Thank you. It's a wonderful talk. So if people have questions, just type your questions in the Q&A box.
So now QA box.
Yeah.
Okay, I see 2. I see 2 questions. So I will just answer it. This is about. It's interesting that the in vitro translation system showed RPA32 mRNA produced more protein with the presence of MecciRNA. Is there any stable base pairing between RPA32 mRNA and MecciRNA? What's the possible mechanism that circRNA enhances specific mRNA translation? Okay, actually, I go back to my PPT. I don't here. So actually MecciRNA does not enhance the translation. Actually, the translation actually is decreased, actually inhibited a little bit. But if we see the protein that get exported, this is the mitochondrial level of the protein, okay? So we can see actually more protein get exported into the mitochondria. So there's no sequence like complementary sequence between the MecciRNA and the RPA32 mRNA. So actually it's just facilitate the protein importation from cytosol into the mitochondria. So at least based on the in vitro translation system, okay? So I don't know this. And then the next question is the interaction circBoule with HSP circBoule, okay? Is the interaction circBoule with HSP depend on circBoule translation? Actually, we don't think circBoule can be translated. So so the answer is no. circBoule RNA is not a translational template. Yeah. So was wondering if bidirectional transcription had a role in generation of circRNA. I mean, the transcription, I don't know. The question is jumping. No, I don't. Anyway, so I think now for circRNA biogenesis, actually, it is possible. So, but people generally think like back splicing is actually after the transcription of the pre-mRNA. So it's at that time actually the transcription direction is already decided. So then the back splicing happened on the pre-mRNA. So, but it is possible. It is possible. So how did you discover motifs 1 and 2 in the circRNAs? I think this is talk about the circBoule. Actually, it's actually we analyze the sequences and from fly to human, and then we can see the two conserved site on the circBoule RNAs. Then we did some mutation, point mutation. We showed these two both motifs are required for the interaction between the HSP proteins and the circBoule RNAs. Okay, complementing my previous question.
Hey there. Yeah.
Okay.
For the time issue, how about you answer the questions in the QA box, and we can also send you all the questions by email with that contact information. So yeah, so we have to switch to Xiaoyan now.
Okay.
Yeah.
Okay. Sure.
Okay.
Yeah.
Thank you. It's wonderful talk. Thank you.
Yeah. Thanks.
Yeah, you can stop sharing. Stop sharing the screen, and then let me try to
stop.
Thank you. Yeah, it's nice, wonderful talk.
Stop sharing. How to stop?
Yeah, and that should be on the right. Right.
Okay.
Left? Okay.
Yeah.
Okay. Let me try to share Xiaoyan's. Let me see how to share my screen. Okay.
I saw it.
Okay. So let's switch to the next speakers, Dr. Xiaoyan, Dr. Song Xiaoyan. Dr. Song Xiaoyan is a professor of the School of Life Sciences, also our University of Science and Technology of China. She received her bachelor's and master's degree in biology from Beijing Normal University, and got her PhD degree in biology from the University of Rochester. Dr. Song conducted a postdoc research training in the University of California, San Diego, and she received a postdoc fellowship from the Cancer Research Institute. I think she joined the School of Life Science of USTC at the end of 2012. So for many years, she has been engaged in epigenetic recognition of gene transcription, focusing on the role of and mechanism of three-dimensional transcription recognition and conditioning of non-coding RNAs and three-dimensional genome structures in biology processes, such as brain aging and somatogenesis. So let's welcome Xiaoyan. So you can start and just tell me, go to the next slide.
Okay, thanks, Shihong, and thanks, Shihong, and thanks the organizer to give me the opportunity to present our work. So my talk title is The Role of Non-Coding RNAs in Brain Aging. Next, please. So aging is a major problem in both biology and society. So we can see on the screen in 2019, the global ranking of population aging in different countries are already very severe. And WHO, please hit, shows that predict, Shihong, please hit, predict 2050, the population aging will get worse. Next, please. So with age, there is some hallmarks show up, and also age-associated diseases will increase the occurrence percent of this disease. Next, please. And here are some examples for non-coding RNAs that are dysregulated in aging-related diseases such as AD, cancer, and diabetes. Next. So in different organs, brain is one of the first organs start to age. So in human, it is reported that brain and lungs are first to start age in 20s. And our work shown also in mice, brain and kidneys are the most sensitive to age signals. Next, please. Neuron tissues is the next to only testes that have the most abundant and the variety of non-coding RNAs. And it has been shown that many non-coding RNAs are associated with brain aging. However, this association, whether they have real function or not, we don't know. And also there are many non-coding RNAs to be discovered and studied. So next, please. So we aim to find this potential functional non-coding RNAs in brain aging. And we start with RNA-seq of old and young mouse brains. And this RNA-seq reveals some upregulated and downregulated transcripts, including some non-coding RNAs. And the goal analysis shows they related to the neuron function and the neurodegenerative diseases. So that makes sense. So next, please, I'm going to talk about 3 non-coding RNAs we screened from the RNA-seq screening. And one of these I will talk in detail is Synage, named from Synage in H. And I will talk to you about how Synage maintains stability and function of the cerebellar synapse. And this work was conducted by 3 very talented graduate students, and Wang Fei will now a postdoc in my lab. Okay, so we first, next please, we find Synage is decreased in old mice brain, and especially in cerebellum. And we found, next please, next, we found it is conserved from mice to human in terms of its genome location. So here left, it shows that in mice, monkey, human, the synage all exist next to CBLN1, this gene, which I will come back later. And right, it shows that in terms of organ expression specificity, both mice and human, the synage expressed highly in the cerebellum and testes. And next, please. Oh, I'm sorry. Yeah. So by RNA fish and the protein IF staining the specific marker of the Purkinje cells, which is the major neuron in cerebellum cavity, we can see that synage is distributed in the dendrites of the PC Purkinje cells, and also it localized in cytoplasm. Next, so we generate synage knockout mice by CRISPR-Cas9 system, and both RT-qPCR and RNA fish shows that synage is depleted from the cerebellum of the knockout mice. And next. And this synage knockout mice show cerebellar atrophy. So the body weight and the whole brain weight are not changed between wild type and knockout, but both female and male, the cerebellum is smaller and the weight is less for the wild type, for the knockout compared to the wild type. Okay, next, please. Before I go into details, I just introduce you about this cerebellar cortex. So cerebellar cortex have major 3 layers. From outside to inside is molecular layer, and Purkinje cell layer, and the granule cell layer. And Purkinje cell and the granule cells are the major 2 neurons in cerebellum. And in molecular level layer, the Purkinje cells, the dendrites branches exclude to this molecular level layer and form synapse between synapse with the parallel fibers and perform its function. Okay, next. So we notice that the knockout of synage leads to the loss of cerebellum neurons. So again, we use the fish and the protein IF staining with specific markers of different cells. Again, encapsulating for Purkinje cells and GDF10 for the Bergman glial cell. And we used PAC6 for granule cells, but the signal was not clear. So we instead used the whole chest staining of the nuclei to roughly monitor the granule cells, which is tiny and small neuron in the cerebellum. And the quantification shows that both PCs and GCs, the 2 kind of neurons, are decreased in knockout condition, but not the Bergman glial cells. Next. And when we inject the synage by virus in the cerebellum in the knockout mice, this actually can rescue the PC numbers. Okay, next. Next, please. And we also use the transmission electron microscopy to check the synapse, the structure, and we found that both the synapse number and the synaptic vesicles compared to the wild type, the knockout mice have reduced both numbers. And also we use the electrophysiological assay to identify the miniature inhibitory or excitatory postsynaptic currents, which reflect the synapse function. And we can see both MIPS and MEPS, they were reduced in knockout condition. So this means knockout synapse reduce cerebellum synapse numbers and their function. Okay, next. And next, please. So, and overexpression of synapse in the knockout mice can rescue the loss of synapse and synaptic vesicles. Next. And we also use different behavior tests show that knockout mice have reduced motor activities. So left shows the road rot test shows that knockout mice have both lower intensity to fall and the running time, running distance on the road rot. And next. And the balance beam test also shows that the motor activity in knockout is reduced. Additionally, next, please. Additionally, we show that knockout of Synage have reduced learning and memory activity. This is reviewed by the novel objective recognition test. Okay, so we showed all different levels of phenotypes by knocking out Synage. And we know non-coding RNAs can regulate the targeted gene expression in different ways. So we're trying to find out the mechanism, how Synage function in this, what I said, phenotypes. First, next, please. We checked the expression level of CBL1, which is next to Synage, and which encodes a synaptic protein in cerebellum. And it is very important for the synapse formation in cerebellum, especially for the synapse formed between the parallel fiber and the Purkinje cells. So next, we found that both CBL1 mRNA level and protein levels by RT-qPCR and Western both decreased in knockout mice. And to rule out it is because of knocking out the genomic structure, we use the nuclear run-on assay to show that actually the nascent transcript of CBL1 is not affected in the knockout mice. Shown in A. So this shows that the effect of the Synage knockout on the expression of CBL1 happens in the post-transcriptional level. Next, please. And we further found both CBL1 mRNA and Synage have the common target site for microRNA MMU near 3 to 5 3P. And we use the in vitro reporter assay shows that really these target sites are functionally important. And D shows that near 3 to 5P is not function in this system. Next. And also we trying to see whether here Synage function as a circRNA. So we searched Argo published ClipSeq data found that both CBL1 and Synage has the binding site for Argo2. And we further use the Argo2 REIP to show that Argo2 interact with the Synage. And by the in vitro system, we can show that binding labeled mu 3 to 5 3P can interact with Synage and CBL1, and the in vitro mimics or inhibitor can respectively inhibit or increase CBL1 protein levels. So indeed, Synage here is a function as a circRNA to regulate CBL1 mRNA and through Argo2 microRNA pathway. Next. And the overexpression of Synage in knockout mice can react to CBL1 protein levels. So here we can confirm that the synapse function loss in the Synage knockout mice is due to CBL1, the dysregulation caused by the knockout of Synage. However, next, when we compare the phenotype of different knockout mice, we saw Synage knockout mice have more severe phenotype than CBL1 knockout mice. So this shows that Synage regulating CBL1 is only partially account for the phenotype we saw in synapse structure and function. So we are trying to see checking for the other mechanism. Next, please. Through in vivo RNA pull-down MS-SI, we found that Synage potentially interact with HSP90AA1 and LRP1. We further use these two proteins RNA-IP to confirm they indeed interact. So next. And what LRP1? It is a synapse associating protein. It interacts with PSD95, which is a post-synaptic protein. Next. And the LRP1 knockout mice have some phenotype similar, the same as Synage knockout and different as CBL1 mice, knockout mice. So it's perfectly good if these two pathways together account for the Synage function in synapse. Next. So through we are look into how Synage regulating LRP1, and through Western blot we found HSP90 and LRP1 and PSD95 in the whole protein level. They are not changed upon knockout of Synage. However, when we use these three proteins individual RNA-IP to check the interaction with the other two proteins, in knockout conditions, all of these three proteins shows decreased interaction with other two proteins. And the right co-IP experiment with this Synage RNA fish confirmed this data shows that Synage mediates co-localization of these three synapse associated proteins in cerebellum. Next. In vitro, we also use the EMS to show that Synage directly interact with LRP1 and PSD95. And the in vitro transcript Synage sense transcript can increase the interaction between LRP1 and HSP90A1. This data show that Synage function as a scaffold for assembling these three synaptic protein complex. Next. So we come up with this model that Synage can regulate cerebellar synapse stability and function through at least two mechanisms. One, in parallel fibers, it functions as a circRNA to regulate CBL1 protein and mRNA level. And in the other, in PC cells, it's function as a scaffold to orchestrate the assembly of the three synaptic proteins, PSD95, LRP1, and HSP90A1. This account for the function of the synapse mechanism. And this part of the work has been published in CCD. Next, since we are focusing on the aging function, so we also found that knockout Synage leads to the premature aging in cerebellum. So A and B shows that the beta-gal learning in knockout mice increased, and the other part shows that the motor and learning and memory function is reduced in knockout mice. Next. And also, knockdown of Synage in cerebellum also leads to the upregulation of aging-related genes. And currently, we are studying the detailed mechanism how Synage regulating the age, brain age process. Next. So this is Synage. Next, non-coding is method 1. Method 1 is the famous express very high and nuclear localized. So time is almost done. I will be very quick in the following slide. So we found that method 1 is increased in old brain and also in retina and of factory bulb, and also in IPS derived optical cups. Next. And we generate 2 knockout mice that shows, and with the detailed experiment, we show that 2 parts of the method 1, 5' and 3' are independently expressed. And later, next. The next non-coding RNA, and we show next. It's also increased in brain aging, and we generate the double knockout, single non-coding RNA, and protein knockout, the 3 knockout mice. Next. And through different behavior tests, we show that these old double knockout mice show clear more anxiety. So this is shown by marble bearing test and elevated plasmase test. And next. And dot-light transition test. Next. And finally, I will be very quick. I just thank all the members of my lab, and especially the people who are conducting the projects I just talked about, and also all the collaborators. And thank you all for your attention, listening. Thank you.
Okay, thanks for the talk. So we still have several minutes left. So how about people typing their questions in the Q&A box? Yeah. So Xiaoyuan, I think you can see check your Q&A box whether people have directly asked questions there.
I didn't see any question.
Okay, so actually, I have a question here. So about the function of method 1, people have debating for such a long time. So what could be the reason for that?
Yeah, that's a good question. And we have been challenged by all the reviews. So, so there are, before our knockout, there are at least 3 knockout mice, and 1 like 5' knockout, 1 the whole region, and 1 inserting of the poly-A to stop. But all of these studies, they only examined early age of the knockout mice, up to 6 weeks. So this is one reason. And another reason is, I don't have time to talk about, is because the mice in our hands, we show the phenotype is that it did not have the expression of the 3' transcript, which is more conserved. But all these 3 other studies, they have the 3' transcript remained in their system. So this could be the reason.
Okay.
Hello.
Yes, hello.
Hi, so my name is Chen, and I'm from the University of Hong Kong. And Q&A, if you see the Q&A, I posted some questions. Can you see?
Sorry, I didn't see. I couldn't.
Okay.
Can you say it?
Yeah, okay.
Yeah,
I have some 4 questions because that's
okay.
so interesting. So one question is that is Synage highly expressed as much as the neighbor genes? So I think the circRNA needs some higher expression level than the target gene. So if you do the qPCR, then the level is comparable
Yeah.
over.
The level is comparable. Yes, the Synage is relative high expressed.
It's interesting.
Yeah.
And the second question is that you did the nuclear run-on. So does NRO maintain the chromatin structure? So I'm not familiar with this experiment.
Nuclear run-on. How?
Yeah.
So basically, you did do this experiment in kind of live cells, and you use the drug to stop the transcription, and then you use the antibody to pull down the PO2, and then you check.
I see. Yeah. And another question is that does LRP have an RNA binding domain, or can you do a clip with the LRP?
That's a good question. No, we haven't. Yes, we are going to check about that. Yes.
I see.
Because from the in vitro, in vitro in MESA, we saw that they have injection, but yeah, we didn't check. We should. Yeah, thank you.
I see. Yeah, the last question. Yeah, it seems that the Synage is on cytoplasmic long non-coding RNA because it binds to the organelles. So can you use the shRNA to knock down this long non-coding RNA, or can you use the Cas13D to knock down this gene?
We can use the shRNA to knock down. Actually, I have showed the study, the data that we knocked down in cerebellum by the shRNA. Actually, we saw the upregulation of H-related genes.
Yeah. Thank you so much today. Yeah, it's interesting. Yeah.
Thank you for your question.
Hello.
Hello.
Nice talk. I have a question for you. Did you check the expression of Synage in Alzheimer model disease, like mouse or even like human brain? Did you have the opportunity to do that and see if the expression was decreased?
We are very we are really hope that we can do that. But yeah, in the end we only show that it is expressed in the human brain, the cerebellum. But because we had tried very hard to find related disease in human and to see whether it changed, but we couldn't find such brain and the disease. So it will be great if we can find it. Yeah.
Okay, thank you.
Thank you.
Okay, thank you everyone for the time issue. So we have to stop. So if you have more questions, just send an email to Dr. Song or send an email to Dr. San. So I think they can ask that, or you just type in Q&A, they can still say it. So thank you very much. Thank you. It's a nice talk. Thank you.
Thanks, Zhihong. Thank